Welcome to section 9.2, DNA replication. In this section, we'll see how a cell copies its DNA before division, so each daughter cell inherits a complete genome. By the end of this section, you should be able to explain the steps of DNA replication, explain why telomerase is important for chromosome ends, and describe how proofreading, mismatch repair, and nucleotide excision repair correct mistakes. Replication happens in S phase, before a cell divides. The double helix is locally opened to form replication bubbles and forks, and each strand serves as a template to build a complementary new strand. Prokaryotes usually have one origin on a circular chromosome. Eukaryotes have many origins on large linear chromosomes, so replication can finish on time. DNA strands are anti-parallel, and bases pair specifically, A with T, C with G. Complementarity is the reason replication works. If you know one strand sequence, the enzyme can build the matching strand. Also, remember DNA polymerases add nucleotides only to a 3' end, synthesizing in the 5' to 3' direction, which shapes how the two new strands are made. Semi-conservative means each daughter DNA molecule has one parental strand and one newly synthesized strand. The classic Mezzelson-Stahl experiment confirmed this by labeling old DNA, then showing that each new double helix carried one old strand paired with one new strand. This model helps explain both fidelity and continuity of genetic information. There are 3 stages of DNA replication. The first step is called initiation, and it is where proteins recognize origins. Helicase unwinds the helix. Single-strand binding proteins stabilize exposed strands, and topoisomerase relieves twisting ahead of the fork. Next, in the elongation step, primase lays short RNA primers. DNA polymerase extends from primers in that 5' to 3' direction. The leading strand is continuous. The lagging strand forms Okazaki fragments that DNA ligase later joins. Finally, in the termination step, primers are removed and replaced. Ligase seals remaining nicks to complete the sugar phosphate backbone. On linear chromosomes, DNA polymerase can't fully copy the extreme 3' ends, so some sequence is lost with each round of replication. Telomeres, which are repetitive non-coding caps, protect genes from this loss. The enzyme telomerase solves the end replication problem by extending the parental 3' ends in certain cells, like germ cells, stem cells, and some immune cells. Elizabeth Blackburn, with Carol Greider and Jack Szostak, helped discover telomerase, and that work was recognized by the 2009 Nobel Prize. Telomere shortening is linked to cellular aging, whereas many cancer cells reactivate telomerase to keep dividing. DNA polymerases proofread as they synthesize. If a wrong base is added, the enzyme senses the mismatch, backs up, excises it, or cuts it out, and inserts the correct base. This 3' to 5' exonuclease activity drops the error rate dramatically. You can think of it as autocorrect for DNA copying. If a mismatch slips past proofreading, mismatch repair scans the new DNA for small mispaired bumps, removes a short stretch containing the error, and fills it back in using the old strand as the template. In bacteria, the new strand is recognized by transient lack of methylation. In eukaryotes, repair proteins use nicks and other cues. Timing is key. This happens soon after replication. Nucleotide excision repair removes bulky lesions, like UV-induced thymine dimers, that distort the helix. A multi-enzyme complex cuts out the damaged patch, DNA polymerase fills the gap, and ligase seals it. When nucleotide excision repair fails, ultraviolet damage accumulates, highlighting why sunscreen and repair systems matter. When the DNA damage response works, errors are fixed and chromosomes stay stable. When it fails, unrepaired DNA can become permanent mutations. Some mutations are neutral, but others disrupt gene function and can contribute to disease, including cancer. Cells balance speed and accuracy to copy billions of bases reliably. In this section, we've seen how DNA replication ensures that every cell gets an exact copy of the genetic material. The process is called semi-conservative because each new DNA double helix has one strand from the original DNA and one newly synthesized strand. Replication starts at origins and proceeds through three main stages: initiation, elongation, and termination. During elongation, enzymes like helicase, primase, and DNA polymerase work together to copy the strands. On the leading strand, DNA can be synthesized smoothly, while the lagging strand has to be built in smaller Okazaki fragments that are later joined together by ligase. Accuracy is critical. DNA polymerase checks its work, and additional systems like mismatch repair and nucleotide excision repair step in to fix mistakes or damage. When these systems fail, mutations can occur, and in some cases, this can lead to diseases like cancer or genetic disorders. Finally, we looked at the role of telomeres and the enzyme telomerase. Telomeres act as protective caps at chromosome ends, and telomerase helps maintain them in certain cells, such as germ cells and stem cells. Without this system, chromosomes could shorten too much after many rounds of replication. Altogether, DNA replication is a highly coordinated process that balances speed, accuracy, and stability, and it's one of the most important reasons life can be faithfully passed from one cell generation to the next.